So just so you know that we're recording this, and we'll share that on our website for future reference. So without further ado, I'm going to now hand the microphone and the video over to my colleague Lutz Maribold, who is the head of our Mazingira Environmental Research Center here at ILRI, to start off the series of presentations. Thank you, Lutz. You can see our Mazingira Center in the background there. He's missing it.
Very much. Thank you. Thank you, Polly. Good morning, everyone. And as Polly mentioned already, we're doing this the first time virtual. So if we might have some hiccups with some technical issues, kindly bear with us, but we will try our best, and we tested a few things in advance. Just a few more things. We're not going to bore you for 2 hours with presentations or something, even though we are used to doing such workshops or something in a more sort of physical way and also with some practicalities. We moved virtual and we tried to boil things down to some key messages. And so we're having just very short presentations, and there will be plenty of room for discussion later on. So today it's the first of 3 seminars we're giving in these coming 3 weeks, and today we are just dealing with mitigation. Next week will be adaptation, climate change, and the 3rd week will be about future scenarios. So let's just start right away with greenhouse gas mitigation in agriculture. And I just want to show a little bit of what we try to achieve today, and I hope this is really going to work. We would like you all to know and to understand the sources of greenhouse gases in the livestock sector. We'd like you to know about different approaches and the quantification of greenhouse gas emissions from manure, animals, and soils. We would like you to be able to evaluate the differences between greenhouse gas emissions and greenhouse gas emission intensities, and I will share a short figure on that later on. And we also would like to go as far as that everybody is understanding on how observations, point scale observations, can be scaled at some point. And now I want to, here we go. So what is our agenda for today? I will do a rather brief introduction on greenhouse gas emissions from the livestock sector and a little bit on the terminology. And then Sonia Leiter, she's a scientist in our lab, will talk about manure emission measurements, tier 2 and tier 2 and tier 1, which will be followed by animal greenhouse gas emissions from 2 colleagues from the lab, scientist Venya Marquardt and our PhD fellow Phyllis Ndungu. And then Mike Graham, who is a postdoc in the lab, will talk a little bit about scaling and greenhouse gas emission modeling. So what we're going to do is, if you have questions, we kindly ask you to really raise those in the chat. And we will always take time for 1 or 2 clarifying questions after each presentation. But we do the whole full Q&A after the presentations in total. And we have Laura and Polly sort of monitoring the chat so that we are not missing any of those questions. All right, so now let's start. What do we know? And when you go back a little bit, this is a study from 2014, and you look at the agricultural sector and greenhouse gas emissions by source, you see that actually agriculture emissions are dominated by livestock emissions to a certain degree. And I mean enteric fermentation, but also manure management, and manure applied to soils. And when you look at the recent modeling studies, we know about sub-Saharan Africa, that agriculture in total contributes between 10 to 80% of the anthropogenic greenhouse gas emissions. It really depends on the country you're looking at. And of these emissions, more than 70% are usually originating from livestock, because livestock is an important asset in agriculture in sub-Saharan Africa. However, all these numbers are actually, to a certain degree, based on guesstimates from intensified systems in developed countries. And we actually do need localized evidence on production rates in livestock systems, and environmental consequences need to be quantified. Why is that important? Well, basically, with this information, all the individual countries are actually able to fulfill the commitments made under the NDCs. But also, if we have a better understanding of the systems in reality, we can think about targeted interventions to achieve climate-smart livestock systems. meaning we can try to boost productivity, we can try to reduce emission intensities, also be better adapted to global climate change. As mentioned, these numbers as well, they're sort of guesstimates to a large degree. And that's the reason why I would like to go back just one step and explain the tiers to you. So current estimates, and this is really for the majority of cases, the reality, are based on a tier 1 approach, which is the most simple approach on how you can actually derive greenhouse gas inventories at a national scale. They're based on an emission factor, meaning very simply on how much methane, for instance, a cow emits per year, and the number of animals. These emission factors are built on best estimates we have or guesstimates we have at the moment. And just by multiplying the emission factor with the number of animals, you get a greenhouse gas inventory. The next step would be the tier 2 step. It's a little bit more sophisticated. You still use emission factors, but they are based on localized data, on the reality you find, let's say, in Ethiopia, in Uganda, or in Kenya, accounting for feed variations, for seasonality, because you have wetland by season, for breeds, and for production systems. And you also contain more detailed information on the actual herd structure of how many female cattle, how many male cattle, how many cows you have. And last but not least, the most complex approach is the tier 3 approach, which uses the original tier 2 values with the localized information, but puts these into an ensemble of several models. With that, you can actually run mitigation scenarios, but you can also continuously track your emissions. Now here they look very separately, the 3 approaches, but overall it's more a continuum. So no country is moving from one day to another, from tier 1 to tier 2 or tier 3. It's just sort of a progress, basically. Every time you have more data, you're moving from one to the other slowly but gradually. The second thing I really would like to highlight, and I want to walk you through this, is the difference between greenhouse gas emissions, greenhouse gas emission factors, and emission intensities. Very often these three terminologies are mixed up, and I just want to try to give you a simple example. So if you see this figure, you have it's not dying as below 100%, but it's also not producing. And I highlighted here, anything below is actually a cost because you're only having an animal, but it doesn't give you anything. And as soon as you go above these 100% of your energy requirements of an animal, you start to produce either milk or meat. When you relate this to methane, we know very well that above these 100% feeding, let's say 200%, 300%, that methane goes up, similar to your productivity, but at a lower rate. And that's the reason why I showed you methane here on the right side. So anything you see here for the 20, let's say, kilograms of methane, this is our actual greenhouse gas emission, okay? This is a fully, it's a quantified measure of how much greenhouse gas emissions you're producing. If you're going a bit further and look at the ratio between our methane we are producing and the productivity, here 20 divided by 30, it gives us a value of 0.6. We can go down this line to the very beginning, and you see that the numbers are actually getting bigger. This is our greenhouse gas emission intensity. This is really important to say that. So overall, with higher production, our total greenhouse gas emissions go up, but our greenhouse gas emission intensity is actually getting smaller. We can go a bit further, and what actually happens below these 100% feeding, actually animals try to digest all the feed they can get. And thus, actually, we just recently found out that methane also goes up. We did one of the first studies of that in East Africa. And I think more importantly, most of the farms in East Africa are actually operating at a relatively low level, which already shows us that there's quite a potential to actually increase the productivity in these systems. So to summarize, a greenhouse gas emission is an absolute value, let's say kilograms of methane from a cow, or kilograms of methane or nitrous oxide from a manure heap. A greenhouse gas emission factor is an absolute emission over time. Okay, so how much does an animal emit in terms of methane for, let's say, 1 year. And Phyllis and Svenja will say a bit more about this later on. And our actual greenhouse gas emission intensity is the greenhouse gas emission per product, so not over time necessarily. And those are the values which actually go down the higher the production we have. This is just a little bit on the terminology, and I'm just having 2 slides on Mazingira. Some of you know the Mazingira Center already, others may not. It's a state-of-the-art environmental research laboratory hosted by ILRI and operational since the end of 2015. And we have 3 core mandates, or we have 3 visions. First of all, we would like to provide crucial environmental baseline data for agricultural systems, of course, livestock system, and environmental data are often greenhouse gas emissions. We are also aiming at identifying and testing climate change mitigation adaptation intervention strategies, meaning we're really looking at experiments where we can improve the productivity or reduce greenhouse gas emissions. And last but not least, but similarly important, we want to serve as a center for capacity building for the next generation of scientists, technicians within the environmental sciences domain in East Africa. And that's the reason why we do have quite a lot of master and PhD students. The team is larger than 30 and consists of scientists, postdocs, technicians, and assistants. So what have we done so far since 2015? We have generated tier 2 manure management emission factors for smaller livestock systems in Kenya. And these numbers are also can be found in the refined guidelines for greenhouse gas inventories by the IPCC, updated in 2019. We have developed some tier 2 emission factors for dairy cattle in Kenya, which are already used in the national greenhouse gas inventory. We have looked at several animal interventions. We also looked at greenhouse gas emissions from land cover types. But besides just providing data, we also produced a lot of training materials and feedback workshops for farmers. If you really want to know more in detail, and all the data and publications are available, please check out our webpage. With that, I would like to close my first session here. And there's time for about 1 or 2 clarifying questions. And otherwise, if there are none, but we wait a couple of seconds, we will move on with Sonja. Okay, since there don't seem to be questions right now, I would like to hand over to Sonja.
Good morning, everyone. Thank you very much for joining us. Let me share my screen here in a second. Just 1 second. My name is Sonja Leitner. I am a scientist at ILRI's Mazingira Center, and I work a lot on soils and soil greenhouse gas emissions and emissions from manure. And I will talk to you a little bit about how we can estimate manure and soil greenhouse gas emissions and why that's important. Okay, seems like my computer is hanging a little bit. Okay, here we go. So you've just seen this figure from my colleague Lutz. These are the greenhouse gas emissions from the agricultural sector by source. And if we sum up all the emissions that are related to manure and manure management, you will see that 25% of the global agricultural greenhouse gas emissions are related to this sector. Now, if we look at the tier 1 assumptions, the basic assumptions from the IPCC for cattle in sub-Saharan Africa, you will find that the basic assumption is all manure is left on pasture without management. And the default emission factor for manure in sub-Saharan Africa is 1 kilo methane per animal per year. But of course, we do know that there is a lot of different, there are a lot of different manure management systems in sub-Saharan Africa. And these systems depend a lot on livestock system and on animal confinement. So when we talk about animal confinement here at ILRI, we have a very basic classification system. We group it into 4 different groups. The simplest system is the grazing or pastoral system, where we basically have no confinement at all. The animals are left on pasture for most of the day and can roam around freely. The next one is the fence-only system. This is basically in Kenya known as bomas, or there's also the term corral or kraal. Basically, these are mobile fences that can be set up temporarily. They're often set up overnight, and the animals are confined against theft or wild animals, for example. And this is the next step of confinement. And then if you look at the 2 pictures at the bottom, we have fence and roof, and fence, roof, and floor. And these systems you can find in a little bit more intensified livestock systems. You will often also find more improved breeds, crossbreeds, and also animal feeding becomes a little bit more advanced and animals start to get supplements, etc. Now, what does this all mean in terms of manure management? If we have a grazing system, most of the manure will remain on the pasture where it is dropped. And it's mostly not used at all because it's very difficult to collect. If we have a fence-only system, some of the manure will remain on pasture when the animals are out during the day, but a lot will also accumulate in the bomas, in the corrals overnight. And sometimes you already find some sort of manure management here. Sometimes the manure is collected and sold to farmers, for example. Very often it remains in the bomas. If you have more intensified systems that have a roof and a floor, you will find a multitude of different manure management systems. The simplest and most common one is the manure heap, but there are also farmers that use biogas digesters, and very often manure becomes important as fertilizer in these systems. So for us, the first step when we want to learn about manure greenhouse gas emissions, we want to know about the management and we want to know about the confinement system. And this is something that we frequently do when we have studies in the field. So this is one example of a study that we did in Tanzania 2 years ago, where we did household surveys on animal confinement and manure management systems. We did this in 2 districts in Tanzania with a couple of different households participating. And we asked the farmers about the confinement systems and manure management systems in place. And on this figure, you can see that we also stratified the data based on agroecological zone. So this is based on the climate, on temperature, precipitation, and elevation. And here in this figure, the yellow and gray areas are the midlands. And you can see that in these areas, most people use the fence-only system. And the blue area is the highlands. And here a lot of people already had fence, roof, and floor system. And in terms of manure management, the most important management were solid storage and the dry lots, the bomas. So this is basically the first step, how to estimate emissions. You ask surveys about confinement and manure management systems, and then you can use tier 1 emission factors to estimate greenhouse gas emissions. But sometimes we will find that the emission factors that the IPCC reports are not valid for African systems. So we want to come up with our own local emission factors. And I want to present to you 2 examples of how we did this in Mazingira in the past. So this example here, we wanted to know what the greenhouse gas emissions from manure from local cattle versus improved cattle are. We did an animal feeding trial in Nairobi in the Mazingira Center, where we used local Boran cattle and imported Friesians. We fed them 3 different diets, a basal diet and 2 improved diets where the animals received more protein. And then we just applied the manure to the grasslands and measured greenhouse gas emissions. And I don't want to get too much into detail here, but what we found was that there were differences in nitrous oxide emissions from manure between the different breeds and also across the diets. And we also found that the emission factors that we calculated, which is N2O, applied manure nitrogen emitted as nitrous oxide nitrogen, so how much of the nitrogen that was in the manure ends up in nitrous oxide. These numbers were much lower compared to the IPCC default emission factor. And this was related to the poor quality of the cattle manure, even in the improved diet. And in this case, these numbers are now also part of the updated 2019 refinement of the IPCC guidelines for national greenhouse gas budgets. So this is one way how to estimate and how to produce your own emission factors. And then we wanted to know, is this also the case if we don't look at manure on pasture, but manure in heaps? So we did another experiment where we looked at the effect of dung quality on greenhouse gas emissions from heaps. We did an animal feeding trial with cattle again, and you just heard about metabolic energy requirements from my colleagues. So we fed the animals at 3 different levels of metabolic energy requirement, 100%. That is basically where we were meeting the basic requirements for the cattle to basically maintain weight, but they didn't grow or they didn't lose weight. And we also fed them below their energy requirements at 60 and 40%. So those were some really hungry cows. And we collected the manure from the cattle and incubated it in heaps, which you can see on the right-hand side. And we constructed some mobile removable greenhouse gas flux chambers. These are the silver boxes in the picture, which we could put on top of the heap temporarily to collect greenhouse gas samples. And again, we found that emission factors for methane and nitrous oxide were lower than IPCC Tier 1 default values. And also this was again related to manure quality. And in this case, we found that the hungry cows produced manure that was of lower fertilizer value than the cows that received more feed. So in conclusion, we know that greenhouse gas emissions depend on livestock confinement and manure management system. So the first step to estimate these emissions is to classify the systems. And we also know that systems in East Africa can be quite unique and diverse. So the Tier 1 IPCC assumptions and default values might often not be valid here. So we need localized in situ measurements to produce our own data. And these measurements should consider breeds, local versus improved breeds, feed quality and quantity, manure storage type, and also think about other use, for example, if manure is burned as fuel, for example. And of course, we need to cover different livestock systems and animal species. I presented here some results from cattle, but the same can be done for sheep, goats, etc. And we have to think about our ecological zones, climate, etc. And the first step to achieve this, of course, is capacity building. So first you need to know about the systems, and then you can ask the right questions. And I know that not everyone has a full greenhouse gas measurement laboratory at their hands. But I want to assure you that a lot can be done already without a laboratory. And also this is a part where where we also want to see one of our strengths in collaborations with external partners. And a little bit of literature resources. So we can also share this with you, the links with you afterwards. We recently published a protocol for Tier 2 emission factor generation for cattle manure. This is available online on our website, and it's open source. And also all the publications of our studies are available freely on the website of the Mazingira Center. And with this, I would like to thank you for your attention. And I think we have time for 2 short questions in case there needs to be any clarification. And otherwise, I hand back to my colleague Lutz.
Thank you, Sonja. And just to summarize quickly, you should have gotten an idea of what the difference between Tier 1 and Tier 2 emission is from manure, and how these numbers can actually be generated. And the good thing is quite a lot of numbers have already been generated and are available. And if you need some of the data, just let us know and you can contact us. One thing I would like to point out, it's not just purely about emissions or emission factors, as already said by Sonia, but also when you think about manure, it is a valuable resource for farmers in terms of fertilizer. And again, we have shown quite a good example on how poor the manure gets if an animal is not fed properly. So I think there's a nice angle for deriving a climate-smart livestock system in the future. And I see one question from Solomon. What is the magnitude of the difference in greenhouse gases using tier 1 and 2? Does it worth the cost of specific estimation? Sonia, do you want to answer or shall I answer?
Yeah, so thanks for the question, Mr. Solomon. It's hard to tell you like really a precise number on what the difference in magnitude is. But let me reshare my screen here really quickly to answer this question. So here, if you look at the left-hand figure, the emission factors, you can see that the IPCC default is 2%. And what we found was below 0.5, so 0.5 to 0.25. So this would mean that if you use the IPCC default emission factor, you will overestimate your N2O emission by a factor of 4 to 5. So you can see that this can make quite a big difference. Of course, it will depend which systems you have, if you have more pasture-based systems, or if you have a lot of different manure management systems. But I would say, yes, it makes a big difference. Of course, we also have to look at where we are in terms of the data availability. And one thing that I would also like to point out, that there is not, it's not that you do either tier 1 or tier 2. There is a gradient between the two. So you can already calculate something like, we sometimes call it improve tier 1, where you still use default emission factors, but you get better numbers on your herd structure, on your animal numbers, on the animal feed intake. And you can use this in the IPCC calculations to get better numbers. So even if you don't really measure greenhouse gas emissions from the manure, you can already start a lot by improving the underlying data. And this will improve the quality of your estimates. Thank you.
Thank you, Sonia. There have been 2 more questions from yours. Why did we use important Frisian and not local Frisian? Actually, that is a mix-up. It was actually a local Frisian bull. So just to clarify that. And the other one you had on feeding maintenance energy requirement, how did you estimate the animal's DMI? This was actually done all in the laboratory. We took the feeds, we did the whole wet chemistry, etc. And one more from Bernard, I'll quickly answer that. Are the manure emission factors applicable for all ruminant livestock, since the research has been done on dairy cattle research? Sonia, please go ahead.
No, I wouldn't say that. So you can probably simplify if you put ruminants together in one box. But for example, if you think about other species, monogastric species like pigs or chickens, the magnitudes will become very different. So I wouldn't feel comfortable to use a cattle emission factor for pigs, etc. So I think it really makes a difference because the animals are different in terms of their nitrogen efficiency, in terms of the water efficiency, the manure chemistry is different. So no, you cannot generalize this across all species.
Okay, thank you, Sonia. Thank you for all those questions so far. Please continue writing your questions in the chat and we will come back to those if we haven't answered them yet. With that, I would like to move on and give the word to Phyllis Ndungu, who is a research fellow in our lab, and she will tell you a little bit about her experience in the field and how to derive animal greenhouse gas emission factors. Phyllis, you have the floor.
Hi, everybody. So I'm just going to share my screen. Yes, so my name is Phyllis Ndongo. I'm a PhD fellow at Mazingira, and my research focuses on greenhouse gas emissions from livestock sector. So our learning goal for this session will be to know and understand the techniques to estimate enteric methane emission factors and estimate emission intensity from livestock sector under field conditions. And we are going to focus on the IPCC Tier 2 methodology, and to estimate enteric methane, daily feed intake data is needed. However, this data is not locally available or is not either updated, not available, or at some point is limited. And this is mainly because most of the systems here in Africa are particularly for under the extensive grazing system, and therefore most farmers don't really take that, are not keen in weighing how much the animals eat. And so to do this, our aim is to estimate how much feed intake these animals consume. And so we go the long way where we collect data such as weight gain, average weight gain, meal production, work or distance traveled during grazing, and other hard parameters such as percentage of females that give birth in a year, and also other data such as digestibility of feed. And then again, in many countries, we're having different regions which have different sub-regions, or we can call them agroecological zones. And these agroecological zones, again, like Sonia said, have different in climatic conditions such as temperature, precipitation, and all that. And that again can affect the diet or the feed or feed available and the quality. And therefore, when conducting these studies under field conditions, it's good to have that in mind, so that you can have a variety, so that you can be able to show the variability in all this. So I'm going to explain what happens from when the project is starting all the way until we get results. And usually the first step during the project planning is that we identify a study site and then inform the relevant authority, and then they asking for consent. And once they give us the consent to go ahead and do this project, we then do sampling. This is done by ArcGIS experts, where they sample farm households randomly. And then once we have those samples, then we visit these farmers, explain what we are doing, and then ask for their consent. And once that is done, then the next step is usually to now collect the actual activity data, which in this case is, like we said, we are going to collect weight data. We also have the hard performance parameters. And so we usually collect weigh animals using an animal weighing scale, like seen in the picture number 2. And again, as you can see that the activity is usually very interactive. So when getting the farmer consent, it's very good to also inform them the magnitude of the work that is going to be done, so that they also are fully aware of what is required of them. And again, also inform them of the period, because we collect data for a period of 1 year, so they're also aware of how much we are going to require them and how much you're going to interact with them. So we weigh the animals using an animal scale, and then we also collect hard-cut measurements. And then for ease of recording, because most of the animals don't have identity, we also tag them so that we can be able to keep records. And then we also determine the age using dentation as seen in picture number 5. And all these activities usually, we also interact with the farmers and train them on how to do these things on their own as well. So we also do one-on-one trainings with the farmers during the data collection, which is also a good thing. And then we also collect data on daily distance walk, because again, most systems in Africa, they practice the extensive grazing system, and so many animals do walk in search of feed. And this is sometimes even they walk more distance during the drought season again, and again that data is required so that we can be able to estimate how much energy they use when walking. And then the next is the productivity data, which in this case is milk quality. There is also growth as well. I did mention average weight gain, and that also is done and can be housed in the productivity data. And we do weigh animals on an interval of a period. It depends on where you are, but we try to coincide them with the weather seasons in a particular country. And once that is done, then we usually collect data at the beginning of one season, which is also the end of another season, and weigh the animals again at the end of the season, so that we can be able to see whether there are any seasonal fluctuations that are in between, and that could be caused by maybe feed availability or quality. And then again, there's also the milk quantity and quality, which is also affected by seasons. Sometimes you have seasons with no feed, and cows tend to reduce their milk. So we train the farmers how to keep records. We offer them measuring cards, which are uniform, so that we can be able to also have quality data for all the households. And then once that's done, at some point we collect the milk sample so that we can also determine the quality of the milk for each household. We also I mentioned about digestibility of feed. So in this section, we collect feed, we do a few things, such as determining the pasture yield. Again, the systems commonly are grazing systems, so we also estimate the pasture yield. And in this case, we set up exclusion cages with known area, as seen in the picture number 1. And then we reinforce them because animals tend to hit them and eat the grass. And then after that, after a period of time, for example, 3 months, the grass is cut and then weighed. Some sample is collected for lab analysis. And then other than pasture, we have other systems in between there, such as cut and carry systems, where we have animals being offered other feed other than grass or pasture. And so we also identify other feed stuff available, such as fodder and forage crops. And then there is also crops byproduct, such as maize stovers, and in other farms, they also offer concentrates. Again, these feed stuff are also available seasonally. Like, for example, the crop byproducts, you find that, like, for example, maize stover are only available during the harvesting season, and then when planting season, there is no more. So again, we tend to we identify the availability of these feed stuff and in which season, and then we create a feed basket, which is different for each season, but then we also have the quality of that feed basket for all those seasons. And again, areas are different. So we have different areas with different feed availabilities. Like, for example, we have areas where there is no sugarcane growing. So in those cases, then you don't have sugarcane tops in the feed basket, but in another area or region, you tend to find there is that. After collecting all that data, then there is this flowchart here, which shows how we use all that data to predict how much of feed intake the animals consume. And once that we predict the feed intake, then we go ahead and now calculate how the dry matter, I mean the daily methane production, and afterwards we convert this to emission factor which is a kilogram of methane per head per year. So we have a few results from, we have 2 case studies that we have successfully been able to finish. And in those case studies, we have Nandi and Nyando. And it's good to note that these 2 regions are located in the western part of Kenya, and they're very close to each other. But then what we appreciate here is that you have Nandi having much heavier animals, and again, the emission factor being much higher than in Nyando, which has lighter animals and much lower emission factors. So, and this is what puts weight to what we initially say, that it's good to identify, it's good to have this, to note that regions can have different animal performance, and that can affect the emission factors. Again, to compare that with the IPCC default, the tier 1, like Lutz indicated there, we see that the IPCC in some cases can underestimate the emissions, and in another case can overestimate the emission, and hence the importance to have these localized data so that we can see the variation between regions and again between households as well. So we have open access publications for the 2 case studies here, which will also be shared and can also be found in the Mazingira website for further reading. So the next, the last section is to estimate emission intensity. We have a case study here from Nandi, and in estimating emission intensity, we first identify, we have to first define the system boundary. In this case, we confine ourselves to emissions which are in the farm only, and once a product leaves the farm, then that is catered for another approach. So, and that is what we call the cradle to farm gate. So our system boundary here is the farm gate, and then so we confine to all the emissions that are in the farm, and then in that farm, then so we have emissions such as enteric methane, manure from what Sonia has talked about, and then from feed production. And then we also, if there are other farm inputs such as fertilizer, herbicides, and animals as well, we also account for emissions for production of all these farm inputs. And once that is that, all the emissions are summed up, and then there, that is the calculation of emission intensity is related to farm output, which in our case we considered milk and meat in most of the systems in Africa, but it can go as far as some non-monetary outputs such as insecurity or insurance, such kind of things. But here we only confine ourselves to edible products. And then once that is done, then we have some preliminary results, which are very interesting. We compared these results with already published data from FAO, and we can clearly see here that our results are quite different from what is reported from Africa. It's much lower, and again, it's very close to the global estimate. And that shows again that our systems are not that badly off, but again, what we also learned is that there is a high variation. We have farms which have high emission intensities, and others have low emissions intensities. But that also says there's still work to be done, but at least we are not badly off. Otherwise, thank you for listening. Any questions?
Thank you, Phyllis. While you think about questions, I do the short summary. So you should have seen that it is actually not too difficult to derive emission factors from the field. Well, it is a bit tedious, and the methodology which we have developed is also available. And at the same time, we are strongly working on actually simplifying this methodology, because we are aware that not everyone can remain in the field for a year and sort of monitor all these farms. At the same time, and I think you should have seen that, there's a lot of data actually needed to quantify emission intensities, but it's the first time we have actually ever done this. And the variation in emission intensities, which was shown by Phyllis or mentioned by Phyllis, is extremely large between different regions. In this case, it was Western Kenya. Okay, any clarifying questions? One we already tried to answer. Phyllis, there's one. Do you also have emission factors for beef, since the production system for Nyando are more of beef than dairy? Would you like to answer that?
Yes. So so what we have right now is the systems that we have in Nyando, in Nandi, cannot really be categorized as beef or dairy, because they're kind of like multi-purpose. So the emissions we have as at now would say are for multi-purpose animals, but we would also, maybe at some point, we can also now start going into specifics such as pure dairy system or beef systems.
Thank you, Phyllis. I see a couple of more questions coming up. For the moment, I would like to suggest that we keep those questions with Laura. She's making a long list for us. And I would suggest that we move on, that you hear a bit more. And I would like to give the floor to Svenja. Svenja, are you still muted by any chance? I cannot hear you.
Can you hear me now?
Yes, now we can hear you,
Sorry.
and we can also see your screen. You just have to go into full screen.
Let me. So sorry, I had some problems. So
No
now you
worries.
should be able to see my screen and to hear me.
Yes, here we go.
Yeah. Sorry, I get the sign that my internet connection is unstable, but I hope you can hear me and you can see my screen. Thank you very much. Nice to be here today with you. And my name is Svenja Marquardt. I am an animal nutritionist. I work for the Mazingira Center at ILRI in Nairobi. And I would like you to introduce you or to show you some of the controlled animal experiments we gain emissions of methane emissions. So as you already heard from my colleagues, there's a lack of baseline data when it comes to enteric methane emissions from tropical ruminants fed on tropical diets and kept under tropical conditions. With Africa, we have the reality, we have dry seasons, and related to this, often restricted intake. So a lack of quality, but also quantity of feed. And what we also lack is measured data on interventions that increase animal productivity and thereby decrease emission intensities. So the gram methane per kg of animal source product, what my colleague Lutz already explained. So there's a need to identify promising intervention strategies that aim to increase the animal productivity by decreasing therewith the emission intensities. What I show you here is from a report, Options for Low Emission Development in the Kenya Dairy Sector. And you see here a list of different interventions. For instance, the establishment of fodder, grasses, and legumes. What you can see in the upper part of the slide. And here on this graph, you see on the left-hand side there is, and on the right-hand side of the graph.
Svenja, can I interrupt? Because I can't see the slides changing. Is everybody else seeing the slides changing?
You can't see it.
I only have your major, the starting slide.
That is Okay. Do you see the second slide now?
No, we can't. Can we do the backup and basically, Polly,
Because
can you
I
share your
click
screen
my second
and then we
slide.
just hear the voice from Svenja? Sorry to interrupt for that.
Yeah, just give me a sec.
Thank you.
Sorry for that, and thank you for letting me know.
Just a minute, everyone. We're sorting out some technical issues.
Polly, I stopped share my screen so you can. I can try it again, but yeah, maybe it's
No,
better,
yes.
Polly, if you do the
Perfect.
backup version.
Svenja, you just have to say next slide then to Polly or something like that.
Okay, I see my presentation now. Yeah, thank you. Okay, Polly, please, one more slide, please. Click. Yeah, now you should be able to see the second slide of my presentation, and now the third one. Is everyone seeing the third slide now? Lutz, can you indicate it's now
Is
working?
that the one with the FAO report?
Exactly.
Okay, then we are back on track.
Okay, thank you, and sorry for that. So I have to I have to start again. So what you see here, hopefully now in the upper part of the presentation, these are, as I said, these are different interventions that are presented in this report, Options for Low Emission Development in the Kenya Dairy Sector on the left-hand side. And these are different interventions. For instance, the establishment of fodder grass and legumes that you see here in the upper part of the slide. There's a reduction in enteric methane emission intensity on the left hand, and the percentage change in milk production on the right hand side. So with the establishment of fodder grasses and legumes and feeding this to dairy cattle, there could be achieved a 80% reduction in emission intensity by a 25% change in modeled for semi-intensive system, the blue color means for intensive system. And this is the whole, or this study is based on a modeling approach. My colleague, Mike Graham, will further on tell you something about modeling and the pros and cons. So this was calculated or modeled with the so-called GLEAM model, that there are certain interventions that by increasing, in this case, the milk production, we can achieve this predicted amount of emission intensity reduction. And there are different strategies they tested. For instance, what is also in the list, what you cannot see here in the slide, control of helminths or measurements against East Coast fever. So the next slide, please. Exactly. So what I showed you in the other slide was a modeling approach. What we do at Mazingira, we do actual measurements under controlled conditions. And how do we do this? The farmer is saying, I know how to milk the cattle, but how do I get the methane? So how do we do it? And this I would like to show you in the next slide. Next slide, please. Thank you. We are using our respiration chambers for measuring enteric methane emissions. They the so-called gold standard when it comes to ruminants, and 3 respiration chambers for small ruminants. And we also have cattle holding facilities and sheep and goat holding facilities, so that we can conduct our controlled experiments there at our Mazingira Center. The next slide, I would like to show you pictures of the respiration chambers. On the left-hand side, you see our small respiration chambers for small ruminants, for sheep or goats. And on the right-hand side, you see the respiration chambers for the large ruminants. You can also see there's a cattle in the respiration chamber on the picture of on the right-hand side. The animal is eating, so we have the animals in there for 24 hours, and the animal, of course, gets feed and water around. So the animal is very comfortable in there. And as you can see, there are also big window fronts, so the animals can see each other, because this is very important also for animal welfare issues. But we also need the animals feeling comfortable in these chambers when we do our measurements. So the next slide, please. Next slide, please. Here you see a figure. We have here the box in the middle where the cattle is in. This is the respiration chamber. And we have inlet air, so air coming into the chamber. We have outlet air going out of the chamber, pumped out of the chamber. We have the greenhouse gas measurement instrument, the laser that you can see on this, on the photo. This is on our control room. We have there our control computer and our equipment. And we can also regulate the temperature and the humidity, so the animal feels very comfortable in there. Next slide. And now I would like to show you 3 different experiments that we either already conducted or that we are planning to conduct. And I choose 3 different experiments to show a little bit the different aims or the different purposes or research purposes or research questions that you can have and where you can use these respiration chambers for answering different kind of research questions. So the first study, this was conducted a few years ago. The results are already published. And the study, or the aim of the study, was to impact different intake restrictions, the so-called sub-maintenance trial. My colleague Sonia already mentioned this trial in her presentation about the greenhouse gas emissions. So as she said, we wanted to simulate a situation of a dry season with reduced quantities of feed available, and we wanted to investigate the effect of moderate to severe undernutrition on the enteric methane production of tropical cattle consuming a tropical diet. And as already mentioned by Sonia, the intakes were restricted to at 40, 60, 80, and the control group 100% of maintenance energy requirements. You can see on the pictures, the animal is feeding, is eating there. This is our cattle holding facility. On the left-hand side, you see an animal on a weighing scale. So the next slide, please. Yeah, thank you. So as I said, the results of this study are already published, and you can find them on our Mazinga website. One outcome of this study was that there was a change in methane conversion factor, YM, so the percentage of feed energy converted to methane, and this increased with decreasing feeding level. This was one outcome of the study. And it shows that improving the feeding of ruminants under nutritional stress has not only the potential to reduce their contribution to greenhouse gas emissions, but also has direct economic benefits for the farmer. via improved nutrient partitioning away from the production of methane towards growth. So the methane, or the percentage of feed energy converted to methane, is a loss for the animal and does not go into the production. Therefore, this methane conversion factor matters, and this was an important outcome of the study. Okay, the next slide, please. This is the experiment that I now want to shortly highlight. Is not yet published. It was conducted last year, finished in January of this year. So we are still in the process of analyzing the samples and data analysis. So here in this experiment, we were feeding improved diets to dairy cows. We wanted to test two improved diets in terms of higher protein contents for their potential to lower methane emissions, methane yields, and emission intensities. And we had 3 different diets. The control diet was a normal diet, or a diet that resembles the conditions on a smallholder farm. Then we had a control diet plus the commercial supplement, dairy cubes. And then we had the 3rd diet where we were testing a local legume and the potential to be used as a supplement because of the higher protein content of this diet. Next slide, please. And what we, the parameters we were measuring were feed intake. We recorded the live weight, the milk yield, the N balance, the methane and nitrous oxide emissions from manure. So to this experiment also a manure experiment was related, and enteric methane production using our respiration chambers. And on the picture here, you can see on the upper picture, you can see 2 animals eating, and the milking is done at this moment. And in this lower picture, you can see the feces were collected, and we were collecting the urine, so the separate collection of feces and urine. And the next slide, please. And the next study, and the last study that I wanted to highlight, this is a future study that we haven't done yet. We are in the planning phase of this study. We were also a bit restricted by the recent COVID pandemic that also changed a bit our time plans. But this is an experiment I would like to mention because it shows another purpose of using these respiration chambers, because here we want to focus on the relationship or the impact of animal health on greenhouse gas emissions. We know that there's often the livestock production systems are partly inefficient, and that one of the major causes for this is the chronic infestation of animals with gastrointestinal tract parasites, which leads to low production rates and high greenhouse gas emission intensities. And in this slide that I showed you in the beginning, where we had these different strategies or interventions that were where the gain in productivity, in this case milk production, and the relationship to emission intensity was shown. There was also one of these strategies was, or 2 of these strategies tackled improved animal health. So we know it from modeling approaches that there is that this is an animal health, or focusing on animal health is an important option to decrease emission intensities, but this was not tested so far in Africa. So we want to test the impact of worm infection on animal productivity, greenhouse gas emissions. Here we focus on enteric methane emissions, where we use our respiration chamber, at manure trials, so we are also looking at greenhouse gas emission from manure, and we are interested in the methane emission intensity. So we want to use 2 different sheep breeds, a local sheep breed, Red Masai, and then more exotic breed, Dorper, that are more respectively less resistant to Haemonchus contortus infection. This is a parasite we intend to use. And we want to work with 2 groups of animals infected versus non-infected animals. And yeah, that is the overview. It's a short overview, and these 3 different experiments that I wanted to show you to give you a little bit an insight of what kind of studies we are doing or we can do with our respiration chambers, and the 3 different aspects. So in one study we simulated the effect of a dry season by restricted feeding. In one study we tested promising nutritional strategies, and in another study we want to focus on the relationship of animal health. So thank you very much, and I'm open.
Thank you, Svenja, for providing this information. And sorry everyone for a little bit of audio, but really Germany, where Svenja is located right now, has a reputation for the poorest internet in Europe. We have seen that many times, so apologies for that. On another note, yeah, we took quite some time to actually elaborate on greenhouse gas emission measurements and productivity measurements for animals, because there's this whole field work side of research, but there's also the lab research where we can really run intervention trials under controlled conditions. And especially the last experiment, which we're planning right now, is really of importance given the current pandemic. And nobody really has looked at both the consequences of reduced health in animals on productivity and greenhouse gas emissions. I see that there were a couple of clarifying questions, and I will quickly answer 2 of them. One for Evans, who is concerned about the mental stress of animals in chambers, which I fully understand this question. Be in short, we do accommodate the animals to the chambers prior to the experiments for several weeks. And the other thing is there's actually windows between the different chambers so the animals can see each other and they're most comfortable. We don't see any stress effects. And as a follow-up question to that from Jos, yes, there's plenty of space for the animals to actually lie down so that they are comfortable because they are in there for several hours. All the more sort of detailed questions I would like to leave for the end. Laura is still making a list, and I would suggest we move to the last presentation, which is then going up in scale and how can we bring these point measurements to a larger area. And I hand over to Mike.
Hello, hi everyone, how are you?
Hi, Mike.
Can you hear me all right?
Yes, we can hear you.
I am about to screen share, yes.
Cool, thank you.
All right, so the screen sharing should be up. All right, it should be full screen now.
Yes, and we can hear you well, thank you. Go ahead.
Excellent. Yes, as Lutz mentioned, I'm Mike Graham. I'm a postdoctoral scientist here at Mazinger at ILRI. My work is primarily on biogeochemical modeling and remote sensing. So today what I'm going to be talking about is so we've had all these presentations about all these experiments that Mazinger is doing, and I wanted to talk more about how do we actually take that data and get to broader scale outputs from that data. So first of all, I just wanted to emphasize that experiments and data collection on livestock emissions and other interventions and agriculture in general are the foundation for all the research that we do. And this is a map on the right side here that shows the locations where Mazinger has their experiments within East Africa. And we generally try and do this for multiple years and multiple locations where possible. But unfortunately, because of resource constraints, these experiments that we do are usually for relatively short duration of only a few years. And even if we can do them for a longer time period, that's not something that can necessarily capture something like climate change over time, which is obviously something we're quite interested in terms of both adaptation and mitigation. The other issue with experiments is space. So it's obviously impractical to do experiments everywhere. So the geographical scope is necessarily limited in terms of where we can actually do experiments. So instead of having experiments everywhere, we have experiments at these locations that you're seeing here. So it's somewhat limited in terms of both time and space. This is where models can be really useful. So for one thing, models are relatively low cost. They can be run just using a computer, but I will emphasize again that actually experimental data are essential for modeling, and I'll show later that experimental data are really what we use to create a model that actually works and to make sure that the model actually works. But they're relatively low cost, so that's a nice feature. They're also capable of simulating conditions, climate conditions, and other environmental conditions related to the agricultural production over longer time periods. For example, if you wanted to do a look at for over 100 years, you could run an experiment as a simulation for that time period. This is also particularly useful for looking at the impacts of future climate change, where we can simulate future climate conditions over time. And models can also be made spatially explicit, and what I mean by that is that you can find out what is going on at a very specific location. So for example, I showed you the map earlier where we just had points on the right-hand side, whereas now you see that the data is spread out everywhere on the map. And this is what we would like to have in the future, to have continuous data across the map at specific locations, rather than just have the individual locations with their experiments. And the map on the right is not something we've done yet. This is just a map of climate zones, actually. So models can be quite useful because they can allow us to look at things over longer time periods and over larger geographic areas. So you've heard a lot about it already, about tier 1 and tier 2 emission factors, and tier tier concept of the IPCC. So oftentimes when we're talking about models, what we're going to be talking about is tier 3 models that have been developed for multiple purposes. So in many cases, these are whole farm models that have models of the entire farm and all their components. You can see on the right here, it has the components, and then you have these processes, and you can measure things related to greenhouse gas emissions, livestock, manure from soils, as well as cycling of nutrients and carbon within the farm. There are other types of models where you can look at, for instance, if you wanted to look at greenhouse gas emissions, you could look at not just the farm, but you could look at the whole supply chain. For instance, if you wanted to say, if you were importing feed, you could look at what is the greenhouse gas emissions associated with the feed you're importing, even though that wasn't on the farm. That's in the supply chain and not the farm. So the two main types of models for interest here are the first type is called the biogeochemical process model, or a BGC model, and that's more of that whole farm type model that I mentioned. The second model is more of the supply chain model, where we can look at things throughout the supply chain and not just on the farm, and those are called life cycle assessment models. So those can kind of give you a bigger picture of not only the farm, but the entire sector, if you want to look at that level. So how do these models actually work? So one of the kind of important things to know about models is, in terms of inputs, they require extensive data to set up, or what we call in modeling terms, calibration of the model. So if you look on the right here, you can see these are the inputs to a model, and these inputs can be quite extensive in terms of the data required. So you need, for instance, data on soil properties, the climate, what are the characteristics, for example, of the livestock herd and its composition, what type of crops do you have, what are the yields of the crops, how about pasture, as well as the climate conditions and things of that nature. So the data requirements can be quite intensive for these models. And in general, some of this data, as I said, will be used to what we call calibrate the model or set it up. But in general, what we will want to do is if we have a large data set from an experiment, so for example, if we're taking one of Mazagira's experiments, we can use some of that data as an input to the model and use it to calibrate the model or set it up. But then we also want to withhold some of that data from our experiments so that we can actually check that the model is working properly, and that's called validation. So we can withhold the results and then verify that the model is actually giving results that are realistic based on our experiments. So it's essentially just the step we do to check that the model is actually working the way it's supposed to, or is similar to what we're hoping for anyway. But there are some limitations to these approaches. So one particular big thing is, as I mentioned, these models tend to require large amounts of data to run if you're running a tier 3 model. Unfortunately, these are typically deficient or in some cases inaccessible in the Eastern African context. So we really do need more geographical data and to be collected, and also more experiments are needed in order for us to be able to properly run these models. So for example, another issue is that different models can give widely different results for the same system. So for example, this plot here is showing N2O emissions from the soil for a specific one farm, and this is for 3 different models. So each of these different types of lines is a different model. So you can see from the red arrows I've drawn here that the emissions, the N2O emissions from the soil that the model estimates are quite varied depending on which model you're using, varying from 0.00 for some models all the way up to 0.03. So one of the problems with models is depending on the model you use, you can get very different results. Another issue is with accuracy. So again, the same chart, N2O emissions from soil is looking at one farm with 3 different models, and the 3 lines are the 3 different models used to try and estimate N2O emissions at that location. I've circled here these dots. So these dots are the actual N2O emissions that were measured in the field. So this was essentially their validation of the model. And you can see that in most cases that the models in this location overestimated the amount of N2O that was coming out of the soil. But I should note here also that N2O emissions are notoriously difficult to model from soils. It's probably the hardest component. So I'm using this as kind of an example, but I don't want you to think that models are necessarily bad at estimating these things. This is just to give you an idea, I think. And in terms of another issue is that there's often, and I'll give you some background on what is needed here, is that their models are often fairly intensive in terms of the skills and capacity you need in order to actually run them. So for instance, you're gonna need a person, if you want someone to work in modeling, that person is gonna have to have a fairly strong background in science and environmental processes in general. So it's kind of useless to be able to run the model if you don't really understand the processes, the fundamental processes you're working with, such as livestock emissions, for example. But you are going to need what you would expect, which is a general knowledge of computers and technology, as well as much more specific skills related to computer programming and coding. You're also going to need to be fairly adept at data analysis, organization, and interpretation. And if you're looking at modeling over larger geographical areas like we are in Eastern Africa, potentially, you want to have some ability to use something like a geographical information system where you're able to do a spatial analysis. I would also emphasize, based on personal experience and experience from what's reported elsewhere, that modeling is not done alone or in a vacuum. These models can be very difficult. There's a steep learning curve, so you're often going to need someone to talk to who is already an expert in that particular model that you're using, and to learn how to use it and to talk with that person or people to figure out how that is done. So with that, I would have a few takeaways and conclusions for you. First, I want to make sure that there's an understanding that models can be powerful tools for upscaling experimental results and other data, but especially in the Eastern African contents, we do need more research and experimental data in order to make our models better and to be able to validate them. They do have some limitations with respect to their outputs, particularly with respect to their accuracy, as well as differences between the type of model you select, the type or the individual model you've selected. And they do require some fairly high inputs in addition to data in terms of the technical skills involved, as well as the resources that you need in order to get these to run. So I think I've given you an idea of what modeling is about, and I think if we have time, I would like to give a little bit of a exercise on the screen here, just showing you modeling can be a bit opaque to a new user, and I was definitely intimidated when I first started working with these, so I wanted to see if we had time to maybe go through a modeling exercise to show a bit of a more simple way of doing this.
Okay, first of all, thanks, Mike, for showing or giving us a couple of insights on modeling. And yes, our plan was not to scare anyone away, and just to sort of give you a little bit of an overview of what is possible and what are some of the necessities. I think we have about 5 minutes for the demystifying the modeling. Do you think that's sufficient?
I had planned for about 10 minutes, so maybe we can do questions instead, or are you thinking that we would have questions anyway?
We will definitely have questions. I'm in contact with Laura. There's quite a lot of them.
Okay.
But I think really, Mike, let's actually move to the questions, because there seem to be quite a lot of them, and let's take a bit of that. Sorry for that, Tom.
No problem.
Okay, thank you. All right, then I would like to hand to Laura, because she has been our bookkeeper in terms of all the questions that have been raised so far.
Thanks, Lutz. Let's see if I can share my screen here. I've recorded all of the questions that have been pasted into the chat, and I've put them on some slides. So let me get that going here. Okay. So let's start here. And then if you have others, please go ahead and put them into the chat as well. I think since I'm sharing now, I won't be able to see the chat. So maybe Lutz, you can monitor if there's others. So Sonia had to leave, unfortunately, but there were some questions for her. I think we answered some already. So Lutz, I'll direct these to you. And then if you want to hand them off to someone else. And I see someone has raised their hand. So let's go through a couple of the questions here. Murayuki, if you're able to type into the chat, that would be good. Otherwise, let's go through some of the questions and then we can come back to you. So the first one on why did you use an imported Friesian in the trial and not a local Friesian bull or cow?
Okay, thanks for asking that, and I think I tried to answer that already. There's a bit of a mix-up. It is actually a local Friesian bull. It wasn't an imported. We didn't fly any animals in or anything. It was something local. So that is a relatively simple one to answer.
Great, and I think we also addressed the second one there, the magnitude of the difference in using tier 1 and tier 2. So unless there's anything else on that one.
I think I would like to add something very quickly on this one, especially on, is it worth the cost of specific estimation? I would probably ask that question slightly different. It's like, is it necessary? And in terms, when you're a national representative for a nation, you are required to actually report your emissions accurately. Yes, so it is a necessity. Now, in terms of cost, there's a lot of opportunity in this, because basically once you have accurate baseline emissions, which we tried to tackle already quite a bit for Kenya, your actually overall emissions might be a bit lower than originally estimated. but it also gives you all these co-benefits, and not just the emission estimates. It gives you these co-benefits of potentials in raising productivity, and by that actually reducing the emission intensities further. So I think it's definitely worth the cost overall. I mean, it's an investment in the future, to say that. And I hope that answered that second question.
Okay. And then the third one here, feeding on ME requirement, how did you estimate the animal's dry matter intake?
I would like to actually push that on to Svenja. Svenja, are you there?
Can you hear me?
Yes.
Yeah, sorry for that. I really have computer problems, but I sorry for that. The maintenance energy requirements were estimated using a regression equation developed from maintenance requirements for non-lactating dairy cattle. And the dry matter intake was calculated by offer minus refusals. So I don't know if this answers this question.
That's fine. That's fine.
All right. And then I think, Lutz, you said you wanted to talk a little bit more about this question. Are the manure emissions factors applicable for all ruminant livestock, since the research has been on dairy cattle research?
Okay, that's a really good one. First of all, we haven't just done sort of the research on dairy cattle only. We have also looked at beef cattle manure emissions, and thus there is sort of different numbers, but between the cattle, I think the differences are not as big as when you think about other ruminant livestock. For instance, we also intend to look at the emissions of manure from shoats, so sheep and goats, small ruminants. And later on, we also we did just a few measurements because it's not the pure ruminant, it's a hindgut animal. We were looking at the emissions of manure from camels. So, and just to add to that, I think what is also necessary is really on where you are located within East Africa and within which system and what the type of manure management is. And I think this is more important than just looking at the difference between beef and dairy cattle. I hope that answers.
Okay. And then I think this one was also during Sonia's presentation. Is the temperature of the locality of data collection important for experiment consideration?
It's a really good one. Yes, it is. And I think it's actually applicable not just for the manure greenhouse gas emission estimates and for any of the surveys we are doing. I mean, the climate itself, temperature and humidity and rainfall is very important because in the end, temperature is a huge driver of decomposition and thus also for greenhouse gas emissions. But overall, I think this is where we link to the stuff what Mike was actually showing, because we are trying to locate our research currently in different production systems, which are then also different, located in different climatic zones. So we're trying to address that to a certain degree. And I think one thing we should possibly also mention here, we used to run also a lab trial. So we have a big incubation cupboards where we can actually control temperature and humidity. We can again sort of look at the responses on how quickly a certain manure decomposes or what are the emissions over time under different temperature and moisture regimes.
Okay. And then what is the variability of estimates in tier 2 within a system?
I think I would like to give this actually to Phyllis because she can answer that very well. Phyllis, are you there?
Yes, I am. So we can have one livestock production system, but again, it does vary because it can vary because of several reasons. We can have the herd structure. You can have a herd structure which has maybe many females, less males, or more growing animals than females, than the adult animals, and that can also can contribute to having variation in a system. Again, We also have, we can have variability in system with, we have farmers who could have the same animals, but they feed differently. They have their different ways, and the kind of feed availability and quality can also be different, and that can also cause variation in systems with the tier 2 estimates. And we have other zones that can be the agroecological zones again, because they have different climatic conditions, and such as temperature and precipitation, which can also have an effect on feed quality and quantity, and that also can cause the variability. So they do vary, and you have seen them varying from worst to good, and there are also others which are in between. So there's a number of reasons that could contribute to that variation.
Thanks, Phyllis.
Yeah, so Phyllis, we have some more for you now. Which parameters of milk quality have relevance to greenhouse gas estimates?
So that's milk protein and milk fat. So if you have the lactose cans, it could be easy to get the milk fat and milk protein with just one test. But in areas where we don't have milk, the lactose cans, then the next thing we do is we use the long method for estimating solid non-fat, because we also need solid non-fat as well. And so we try to get the density of the milk. That is, then we use a formula that is already published to estimate the solid net fat, which is then used. So the parameters required here are milk fat, milk protein, and solid non-fat.
Great, okay. And also for Phyllis, do you also have emissions intensities for beef, since the production systems from Nyando are more of beef than dairy? I think we addressed this a little bit. Do you have any more to add on that?
No, but like I said, it's hard to assign a beef system to most of these systems, livestock systems in Nyando, because again, these animals they're not only for meat, but they also are for milk. Well.
Great. And what is the relationship between type of production and greenhouse gas emissions?
So I can give an example in this case where we, like for example, you have high yield dairy cows, which tend to consume high feed intake. And again, we said we need feed intake data, and feed intake does have a great effect on total greenhouse gas emissions. So having a high yield system would lead to high greenhouse gas emissions, but like Lutz stated in his presentation, we can have high greenhouse gas emissions, but because they're high yielding animals, then we have low emissions intensities. So that is the relationship between production and emissions.
Fantastic. And I think someone found your work really exciting and they want to know what it takes to work with you on the ground. Maybe we can talk with that person individually, but there's some interest there in getting involved in these sorts of measurements. So do you have anything that you want to add on that at the moment?
It would be great.
But we're not giving you that easily away, okay? No, but really, it's a good suggestion to discuss.
Yeah.
That would be great.
Thank you, though. Thank you for the interest.
Fantastic, great. So next one is a fantastic question. I think Lutz, maybe you can take this one. The innovations on methane reduction and increase in production are phenomenal. Do we have mechanisms of linking this research with extension services at the subnational or county level? This is evidence that should inform our adaptation interventions under climate-smart production.
So that's a good one as well. And I mean, this is always what we have in mind. We're not just providing data, but we also want to sort of link to the government stakeholders, but also to the extension agencies. And I think we started that to a certain degree by partnering, for instance, with the Green Innovation Centers from GIZ, for whom we developed trainings, or for whom we already did trainings based on the research we are actually doing. Where we're trying to simplify the research results into some sort of training materials, but also within courses. And I think this is a starting point. Another example is, I mean, and I just elaborate a little bit on this. You saw the importance of sort of a live weight measurement for, first of all, quantifying emissions, but also seeing for a farm on how well an animal actually performs. And not every farmer has a scale or whatever to track this. And that's the reason why Phyllis mentioned we did the scale measurements, but also the heart curve measurements, which is basically the tape around the heart of around the animal to measure the circumference, and then from that you can estimate weight. And we implemented this algorithm in a mobile phone application, so that actually farmers without the scale, just with a simple measuring tape, can track the live weight of the animals, and according to that adjust the feeding of the animal and actually improve productivity. And once and going back to this first simple, relatively simple figure I showed, on where we are located for a lot of East African smallholder farms at the moment in terms of productivity and emissions, just by really ensuring that the animals get sufficient feed during the whole course of the year, that you don't run short during dry seasons, etc., already quite some impact can be achieved. And we are much more willing to work together with other extension services and to really sort of spread the word and really come to this evidence-based sort of advice, which is being translated into practice.
Great. So Svenja, we've got your questions coming up now. The first one is, was quantity and quality of feed intake changed? I think that relates specifically to one of the experiments you were describing.
Svenja, are you there? You're muted.
Can you hear me?
Yes.
Yeah, sorry. In the first trial that I was showing, the example of the sub-maintenance trial, the quality of the feed was the same. It was just reduced, so the quantity offered was reduced. In a second trial that I showed, the improved feeding for dairy cows, yeah, the diets, we had a control diet that was basically hay, brachiaria hay, and for the improved diet with a commercial supplement, we added dairy cubes to the diet on top of this hay. And in the third diet that we had, the local supplement where we used the sodium and hay, the local legume, we added this to the control diet. So the percentage of the brachiaria hay changed. So the basal diet was the same, but to the improved diet we added different components, and the aim was that the control diet has protein content that they can find in smallholder farms, and for these two improved diets we had a higher protein content that was the same for the two improved diets. I hope this answers the question.
Great. Okay, and the next one, I think we talked about this a little bit, but do the animals locked up in the respiration chambers experience mental stress? And if yes, does that affect manure production and emissions exhaled?
So as I said, the animal welfare aspect is very important for us, because we want to measure an animal in normal conditions. We don't want to measure stressed animals, because this would probably create a bias. So we want that the animals have the normal feed intake that they also have outside the respiration chambers. Therefore, we have the big window front, so that the animals can see each other. This is very important, especially when it comes to sheep, that they see each other. And we train or we familiarize the animals with the chambers before we start a trial. The animals enter in the chambers at first for a short moment, then for a longer period, and they really the animals need to know that nothing harmful happens, and that they get their feed, their water, and that they behave normally. And you also see it when you bring the cattle in the respiration chambers. And we do, for each animal, we do 3 measurements. So each animal is measured in each of the chambers. And when you bring the animals in the chambers, they go in there, they start to eat immediately. So this is really, it's a good question. It's really important that the animals behave normally.
Great, thanks. Okay, and the legume used in the trial next to nitrogen, could it have nutritional factors that would influence methane production?
Yeah, that's a good question, and that is also that is true. The legume we use has a certain amount of plant secondary compounds, especially the tannins. The condensed tannins are known to have an effect on the microbes in the rumen. So this is an additional effect that we wanted to test. So this desmodium and totum that we used has these plant secondary compounds, tannins. They have, and tannins are known to have an effect on methane emissions. They can lower the methane emissions. It depends on the dosage, on the type of plant secondary compound, and so on. But this is true. That is something else that was of interest for us, why we were choosing desmodium and totum.
Great, thanks. So the next question, I think you addressed it already. I'll just combine it with the one after. Someone has asked whether there's room in the chamber for animals to lie down. And then the next question is, animals fed on 40% ME, and I'm sorry, I don't remember that acronym. Did they achieve rumen fill, or also dry matter intake was underachieved?
So the respiration chambers are built as such that they have room to lie down, and this is also an important welfare parameter and an important parameter to check if the animal lies down, because this is related to animal welfare, and clearly there has to be room for the animal to lie down. And the second question, yeah, where we had only 40% of maintenance energy requirements. I guess the rumen fill was not achieved. We also did measurements of residence time, and we observed that there was prolonged residence time. But this was due to the restrictions in intake rather than the limitation proposed by physical fill. And I see there's another question for me. Which fodder have higher methane gas emissions, grass or legume? Yeah, we haven't analyzed the data yet. We still have to analyze the data. What we noticed that the legume was not so palatable, so we had a decrease. We had a lower intake in the group that received legume related to the palatability, which might be related to the tannin content. But we still have to analyze the data. But then I'm happy to share the results of this interesting study.
Great, thanks, Svenja. We have a few more. There's one that I think can go to Lutz or to anyone who wants to add. Then there's a couple for Mike, and I've seen a couple more come into the chat. So I'm trying to keep us moving so we can end on time. So this person has said, we are developing a new highly digestible feedstock solution to reduce farmers' costs, improve milk and meat production, and reduce methane emissions. Who can we engage at ILRI to help develop feed application and conduct field validation? Lutz, do you want to answer that one?
Sure, I can answer that. Thanks for pointing this out. This sounds all very promising. Basically, what I suggest is let's have a bilateral chat, probably with me and Polly, etc. And then we can see what we can do. I mean, we're happy to conduct field validations if they're actually feasible. That really has to depend on the setup, etc., and sometimes also a little bit on the costs. But we are happy to help where we can.
Great. And Mike, a couple questions for you. So did you assess emissions from feedlot systems where animals are usually fed ad lib?
I have not done that, but I am not sure if that question is for me. I think that might be, could be for us, a question that Svenja and Lutz can answer.
Yeah, that is, I think, something for Svenja right now. Otherwise, I can support.
We haven't, well, feedlot conditions that you have, for instance, in the US, we haven't focused on these systems. With these trials that these controlled experiments that I showed, they are resembling smallholder mixed system conditions. We haven't gone to feedlot conditions.
Actually, I mean, there is quite a lot of data from, for instance, Australia on feedlots, which could be used as a beginning. And the only thing we have to sort of assess how different the feedlots in East Africa, if you find any, I don't know of any yet, are compared to, for instance, Australia.
Okay, I think that one came in from one of our Ethiopian colleagues, and I think there are some feedlots there that they do some live animal exports, and they have abattoirs for meat exports. So I think I was at a MRV meeting there, and they were discussing feedlots. So that could be something we discuss further with the person who asked directly.
Exactly. Just to add on that, Laura, yeah, you reminded me of that. There's a couple of other countries basically in the Horn of Africa where there are feedlots, and we tried once actually to do a quantification of greenhouse gas emissions, at least remotely as much as it was possible for Somaliland, but in the end we couldn't actually enter the country. But these are all sort of still points where a lot of research can be done and numbers can be actually assessed. Good point.
Great. Okay. And then for Mike, are there initiatives to build local capacity on modeling?
I can speak mainly to what we have in-house at ILRI and Mazingira in particular. So actually Phyllis and Daniel Corrier, another PhD student in Mazingira, are working on some life cycle assessment models where they're looking at the entire supply chain or life cycle analysis to the farm gate for a model for, I believe, Nandi and another location. I'm not aware of any other, and we have another PhD student, Benny Gisore, who will be doing more of a biogeochemical model or a farm model at some point. So those are all kind of in the pipeline. In terms of other initiatives, I'm not aware of any other specific initiatives that are dealing specifically with modeling, but it's possible that if anyone knows of any, I'd be interested in hearing about it, but not that I'm aware of.
Okay.
Maybe to add to this, I mean, there is a couple of initiatives where sort of external partners come in and to sort of promote different models to assess greenhouse gas emissions from specific sectors. And there's also different tools available on direct training activities on these. Usually these are short presentations, etc., but I haven't seen sort of a full curricula or full course on these things yet, but I'm willing to learn about that.
Yeah, so that was, let's mention one of the models is GLEAM, which is a global livestock emission environmental assessment model, I believe it stands for. And we had some people who, before my arrival in Mazingira, came and actually did some training with us. So there's some initiatives like that, but as Lutz said, no courses that I'm aware of.
Okay, thanks. So Lutz, you said you were compiling other questions. I stopped screen sharing. Do you want to take over asking them, or should I start asking from the chat? Oh, great. Okay, here we go.
I think you should see it now, yes. On that first question, actually, I would like to give this to Phyllis, because she has been a lot in the field, worked a lot with farmers, and I think she can best sort of answer that one. Are you there,
Hi.
Phyllis?
Yes, yes, I'm
good.
here. So in the course of collecting data, we do one-on-one trainings with the farmers. So we don't really only just bombard them with all the activities to get the data. We also get to learn to know their systems, interact with them, and then offer assistance where we can, and especially when there is immediate need. But then afterwards, we then, like Lutz said, we offer farmer training workshops where we compile all these challenges and strengths, and we put them in, into a training session where we can now do this further with other experts. Other than that, the interest here is to reduce greenhouse gas emissions. But this also comes with another added advantage where once farmers learn, and like, for example, how to feed their animals in a much better way, then they get to have, once they learn how to do improved feeding, then the next step is they get to have higher production. That means more money in their pockets. But other than more money in their pockets, they then have household welfare is improved. And there's also issues to do with, again, stuff to do with diet as well, our food security. And also the most important thing, which may not be valued, which is information. They get to have information which they can also share with other people who we don't reach. So I believe there's a lot for the farmer, and in return we get to save or reduce the negative impact on the environment, and as well contribute to the welfare of the households. Thank you.
Okay, thank you, Phyllis. Can you all still hear me, because I can't see whether my mic is muted or not?
We hear you.
Good, thank you. Then the second one is, can modeling be done where there is no experimental design, for instance, in transdisciplinary research? I'm not clear if I understand this question correctly, and we might need a little bit of clarification on that. And maybe that can be given in the chat, or the person who asked that can speak up in this case. And while we do so, on the last question, has anyone compiled a list of institutions in the region with reasonable capabilities for greenhouse gas emission studies other than ILRI? What we know of, I mean, there's a few national labs who have sort of some capacity of doing some things. For instance, within Calvo, there is a little bit of greenhouse gas emission measurement capacity. There used to be something within Maseno University in Kenya. There's a recent lab which has been sort of set up in Ethiopia, I know of, together with Norwegian colleagues. And then, of course, there's sort of the typical laboratories within local universities, where especially you have that chemistry and some soil chemistry, which already sort of is quite some good infrastructure in many cases, which can be used and should be used for such research. Besides the fact, and I mean, we can do trainings, etc. So we have a lot of students enrolled with us who get the insights in how to do simple greenhouse gas emission measurements. But in terms of just the greenhouse gas emission quantification, I think we are probably having at the moment one of the most standardized labs in East Africa. If there is sort of interest in getting assistance in setting something up like this, as we have done with Maseno, for instance, we are always willing to do so and share our expertise with that. Laura, back to you.
Thanks. And for the second one here with the transdisciplinary research, Laura Pereira would like to come in. So Laura, can you unmute yourself and give your answer to that?
Sure. Hi, everyone. Good to see you. Well, to hear you. I think Mike also has a comment on this. I kind of think, you know, from the transdisciplinary research perspective, doesn't necessarily mean to say that there's no strict experimental design. It's just a lot more open and participatory. And so I think that there definitely is scope for participatory modeling within this space, kind of working with people to actually see how some different fluctuations kind of work. And so it's less about doing modeling in order to kind of write a paper out the other end, but more to allow people to actually understand what the different processes are, you know, how changing one kind of feedstock has an implication on sort of greenhouse gas emissions and things like that. But so that's sort of my understanding. So I think there is scope. I'm not sure how much of that work is currently being done, but maybe Mike could also talk to it, because I think it speaks a lot to the kind of work that he's been working on.
Fantastic. Mike. Go ahead.
Yes, so my thought on this is, if you're looking at doing a kind of interdisciplinary model where you're kind of integrating multiple disciplines, that's probably something that can be done without necessarily, for example, if you're doing, for instance, integrating an agricultural productivity model with an economic model or a nutrition model or something along those lines. That's certainly something that can be done, and you don't necessarily have to have an experimental design per se to do the kind of more conceptual modeling that Laura is talking about. But I do think you're going to want to have some, if you don't have an experimental design necessarily, you're still going to want to have some data in order to provide inputs into that model at minimum. And that's both in terms of the biophysical part of the model, such as the productivity of the farm, as well as the economics or whatever.
Thanks. Okay. I think at this point, I'll hand it back over to Polly.
Great. That was really wonderful. And I really appreciate the enthusiasm and all of the questions. So as we've said, we will share the recording for this along with the presentations on our PCSL website, and share that with all of you that participated. Just before you go, just a reminder that next week we will be hearing on the topic of adaptation to climate change in livestock systems, and that will be led by my colleague Todd Crane. We'll have the same sort of interactive discussions with each of you, and we really look forward to working with you then. I should say that next week and the following week, we do want to use an interactive survey tool called Mentimeter. You should have received some information about how to download that onto your smartphone, or else how to use it on your computer. So it'd be good if you were all ready to do that next week, because we'll try that out during the adaptation session. Then we'll use it quite a bit in the third seminar, which is around futures thinking and how those kinds of exercises can help us to make decisions about proactively planning for climate change. So if you have questions about Mentimeter, feel free to reach out to Joy or Beth or Tigist, Rahel, Roland, or Sheila, who invited you, because we've been using Mentimeter quite a lot within ILRI, and we can help you do that. Otherwise, I just really want to thank all of you once again. We really appreciated it, and please, if you have feedback for us, let us know. We'll also be asking you some more formal questions at the end of week 3. So thanks again, and I just would really like to applaud the presenters, because that was extremely excellent. I am so impressed with how wonderful those presentations were. So thanks, everybody. Yes, and as people are saying, stay safe and take good care.
Bye, everyone.
Thank you, everyone. Bye-bye.
Bye.
Bye.
Bye, everyone.